The project is affiliated with Ministry of Environmental Conservation and Parks (MECP), public health units of Greater Sudbury, North Bay, Susan Mary, Timiskaming, and Porcupine. Influenza epidemics have been reoccurring for a very long time, even with robust surveillance in place due to longer time frame for the results generated along with the resources used, especially post-COVID-19 calls for more efficient method such as wastewater surveillance, which allowed quicker results. Samples were collected from approved treatment plants, sewershed serving municipalities across northeastern Ontario, which after arrival on sites were extracted using Kaizen Power Microbiome Pro Kit. These samples were analyzed using digital PCR through multiplexing technique. The digital PCR is based on the principle of endpoint PCR, but the PCR reaction mix is split into thousands of single partitions. The nucleic acid template is randomly distributed across all available separations and amplified individually. The amplification target is detected at the end of the PCR process by measuring the presence or absence of fluorescence of sequence-specific DNA probe or intercalating dyes turning PCR digital. As the PCR reaction is partitioned randomly, it is possible that a positive reaction contains more than one target molecule. To account for microreactions with more than one template sequence, Poisson statistics is applied. Additionally, for both influenza A and influenza B strains, limit of blank, limit of detection, and limit of quantification was conducted with non-positive controls. Now, if we move on to results, we can see that 7 sites were inspected for influenza A and influenza B. If we look at the graphs for Susan Mary and Sudbury for both influenza A and influenza B, we discovered that the wastewater spike takes a lead on the clinical data received by 2 weeks. Similar trend is observed for influenza A, North Bay, Timmins, and Kapuskasing, and for influenza B, Timmins and Kapuskasing, with North Bay as an exception where no clinical data were recorded. Moving along, if we take a look at Halberry and Kirkland Lake for influenza A, both wastewater surveillance and clinical data reported shows similar trend unlike influenza B, where PCR assay results are not always backed up by clinical data. Here is the map showing geographical location of the sites being studied. The study so far concludes that for most of the sites, wastewater concentration of influenza A and B backed up by the patient reports. We also observed that the extracted sewershed samples indicates the presence of infection in the population before the traditional test reports becomes available. In some cases, clinical data were not available for the results obtained through the surveillance, which might be because the patient is asymptomatic or affected with the milder infection, which did not result into a clinical visit, and was still captured through wastewater surveillance, providing its coherence. Therefore, wastewater can be used as a tool to monitor influenza among other infectious diseases. The presence of influenza is a leading indicator, wastewater acting as a predictor, potentially due to the shedding of the subclinical cases. Thank you for your attention. Also, please reach out if you have any queries or if you want to learn more about the research work done in our lab through the information provided. Thank you.